All right, welcome to BioSkryb Genomics. I am John Zawistowski. I'm Senior Director of R&D, and I'm joined by my colleague Vic Wigben, who is Senior Director of Computational Biology. And I'm excited to present to you guys today a lens we've put on clonal evolution in breast cancer. And that lens is necessarily single cell, but it's also multiomic in nature. And we do that through a product we call ResolveOME. And for the first time, we're able to unite a complete and uniform genome to a transcriptome in a single cell and obtain insights that we weren't able to obtain in isolation. In addition to talking about ResolveOME, we'll also be talking about BaseJumper, our analysis platform for analyzing and visualizing the complex data sets that we generate with ResolveOME. So at BioSkryb, we believe that complex problems require complex analyses. And we consider ourselves a discovery company, and our goal is to enable single-cell multiomic discoveries. And of course, we have that shared goal in that these discoveries improve human health. So we believe that this requires interdisciplinary research, and I'll give you some examples how we have set that up to answer the most pressing questions in cancer. So taking that core genomic technology, now let's move to ResolveOME. And so ResolveOME is where we've taken the fundamental attributes of this, and we've added a full transcript RNA-seq on top of this from the same single cell. And on the bottom, or excuse me, the left, I'm showing you a genomic coverage graph of how we're approaching that of bulk with our single cell technology. And then I also have sensitivity and specificity metrics for variant calling, which you can see are quite excellent there. And I've already highlighted the allelic balance that we're able to obtain with this technology. So take that genomics and let's couple it to the transcriptomics. And what I'm showing you in both of these data sets are NA12878 cells or HG001. And for example, in that cell line, we can see 4,500 genes detected. And I also give some information about CV and range. And the graph is showing primary samples in a variety of cell lines and the genes detected with our transcriptomics, all in concert with the full genome information. So I wanted to take one slide and dive in a little bit closer on the transcriptomic side. So what I need to emphasize on the left is that this is full transcript RNA-seq data. So in this blue curve, we're contrasting the ResolveOME transcriptomics with N-counting approaches. And so by definition, we have all of that real estate between those two curves to explore with our technology to go beyond N-counting approaches. And this includes fusions, splicing events, and RNA variant detection. The middle graph is showing you this, that already talked about the 5' to 3' gene body, but then this is showing you across transcript size classes. And so ResolveOME technology is on the top, and we have N-counting technology on the bottom, 50 cells that were top performing in the N-counting technology. And you can see with these warmer colors across the transcripts, which are shown in columns, and the single shells are shown in rows, you can see that we have much greater representation across these larger transcript sizes relative to N-counting methodologies. And finally, if we look at variant calling at the RNA level, based on these attributes, we excel at this region as well. So I want to set the stage a little bit chronologically here about the project. And so we first began with about 30 single cells from one patient in this cohort. And we looked at the genomic side, and this was prior to looking at the ResolveOME level. And what we saw to set the stage is really intriguing interplay between copy number and single nucleotide variation in this initial sample. On the left, I'm showing you the actual cells that were faxed. This happens to be ER antibody staining. And one of the strategies that we've taken is enrichment for EpCAM to try to enrich for the ductal epithelial cells in the fax scenario. And then in this first initial data set, we saw quite distinct and striking copy number loss in the tumor biopsy cells versus the normal biopsy cells. I've highlighted some of those with the color with colors showing you not only their genomic location, but some of the prototypical tumor suppressors that are harbored in these regions that are likely contributing to the advancement of this disease. And if you notice, there are classes of this loss. Some of these cells have 2 deletions, some of them have 3, and this manifests throughout our data sets going forward. And the first, and one of the intriguing insights to me at the very beginning is that if we map the copy number alterations in the red text on these initial set of cells, and then we map, for example, a oncogenic driver mutation that we could identify because we have whole genome or exome analysis from these cells, In this case, it's a PI3 kinase mutation, either 1047R or N345K. And as we've done more and more cells, this initial observation in that the copy number loss is linked to only one of the PI3 kinase mutations and not the others has been really interesting to observe. And we're trying to understand why that occurs mechanistically, but I think very thematic going forward, and not only in breast cancer, but multiple types of the CNV and SNV interplay. So as I mentioned, I have one of those icons on the top because we're going to start with the genomic level with this data set. So this is the CNV landscape of all of these patient single cells. And I like to start this way because to me, it's a really intriguing data set where we have 757 cells along the rows of this map. We have coarse chromosome number on the top, and then we have ploidy that is colored by the warmness of colors is the higher degree of copy number increase. And not unlike that first example I showed you, we can see discrete chromosomal alterations across many of these chromosomes, and I'm also intrigued by the degree of chromosome instability that is in this particular cohort. And I've tried to highlight in these dash boxes some of the discrete, even subchromosomal alterations that we can see that differ across patients or between patients. And I've also highlighted with a bracket, we can also look at the quiescent cells. So these are non-epithelial cells. These also are cells that we have for matched normal biopsy. So really striking copy number alterations. And in contrast to that first data set I showed you, many gains that we've noticed as well. So let's again keep a DNA icon at the top of the slide, but let's move from copy number analysis to single nucleotide analysis. So we obviously have genomic information at our disposal. In this case, I'm showing you WGS data where we're comparing normal biopsy cells in blue versus tumor cells on the columns of this diagram, and the variant or the single nucleotide change is shown on as rows in this figure. So we have some of these blocks that emerge for differences in SNVs between tumor and normal due to the copy number losses that I've shown you, that red and black regions. But I'm particularly intrigued by this region in the middle where we have a kind of a classical cancer scenario. We have 0,0 reference genotype in the normal biopsy cells, but we have a heterozygous change in the tumor cells. So what do we see if we go zoom into this region? So in this patient, we see N345K again in PI3 kinase, but other things emerge. So we have NSD2, a epigenetic writer in gene bodies. We have cadherin. We have cyclin T1, which is a transcriptional cyclin, all of which are represented by mutational events, either missense or frame shift in these molecules that can now be exposed at the single cell level. So let's get to the really exciting part. So let's unify these data sets. And so now if we look at DNA and RNA together, let me walk you through this figure. So we have single cells as columns. We have transcripts as the rows. And this is part of a heat map based on gene expression. And what you can see is that there's two predominant clusters. One of these clusters is defined by our EpCAM high cells, and one of the clusters is defined by the EpCAM low cells. And so now I want to walk you through the omic tiers that ResolveOME can provide. So, of course, since it's gene expression heat map, we have RNA information. I've highlighted FOXA1, which makes sense. We're enriched in a EpCAM high and epithelial population versus the EpCAM low population. So let's add another layer. So I've already spoken to you about the nature of the CNVs that we're able to obtain from our data. Now we can, for each of these cells, we can layer that on top and begin to interpret the candidate causative events in these cells. We can then add SNVs. So I'm showing you one of the PI3 kinase mutations, but we have the whole exome or whole genome available to do this for whichever variant of interest that you care about. And I've spoken about the cell ID. So now we can take those EpCAM high cells that we've enriched, and we can see that they also now type epithelial. We can see a diversity of cell types that represent the tumor microenvironment coming out of this EpCAM low population. And the power of this is that the columns of these boxes, and that we have all of this information available for each cell. So as a bullet summary, I've tried to show you our unification of complete genomic, transcriptomic, and now targeted proteomic data to expose insights that really are unattainable in isolation. I've demonstrated this and its utility in primary breast cancer, so CNV, SNV. I've shown you cell state inferences coupled with mutation, and some beginnings of looking at allele-specific expression. And so I want to leave you with what can ResolveOME unveil in your system. Hopefully you've thought about how your system, there might be parallels. But now, importantly, I would like Vic to speak about BaseJumper. And so the data are really complex. The data are large, and BaseJumper is allowing us to gain some of the insights that I've shown you today. So Vic?
So we developed BaseJumper again, really to assess the availability for everyone to draw these insights, right? We looked at it to be easy to use, visually engaging, and fast, right? We want everybody, with no matter the background, to leverage the platform and drive their insights with stuff they're familiar with, right? We want it fast because, again, you can't wait several weeks to get this analysis. You want to understand if that finding you have is meaningful. And it's got to be visually engaging, right? The insights have to come to you without, 5 years of a doctoral in bioinformatics.